Brilliant. Okay, I hope this is indeed working. Please let me know if you can't hear me, alright? But hello again. For those of you who are new here, I'm Henri from the Student Tutors. So I'm an undergraduate student at the University of Oxford and I'm studying biology. And today I'm going to be looking at some questions on cell division for GCSE biology. Like I said in the start, let me know if there's any problems with sound or vision. I'll try and keep an eye out on the chat for anyone who's with us live. And feel free to ask any questions at any time. I'll be sure to sort of go through things a bit slowly just so you have time to think between questions and to sort of come up with your own answer before I go through an answer myself. So, first things first, I guess we'll jump straight into looking at some questions. So, we have here, it says question 2. Question is about cell division. Okay, great, that's what we want to be looking at today. So we've got, which process makes two identical new body cells for growth and repair? So think like growth and repair. Oh, pen's run out a little bit. Growth and repair. And we've been told these are also identical. So it can't be differentiation because that doesn't make new cells. It's one cell turning into another. It can't be fertilization because, again, new cells. So I'm on your exam board. You might know this as cytokinesis. So that's what we know. So if you take a look back, which one of these could be 1, 2, or 3 is what we're looking at. So if we go back here, let me just check the stream's running all right. Seems to be running right. Oh, maybe it's not running all right because it says there is currently no data. Which is interesting because there should be data. Okay, I'm gonna assume this is still running. The clock's still running. It's still registering that I'm recording something. So hopefully it's alright. Again, let me know. I've got the chat open on the side, so if something pops through, I should be able to see it. Okay. So we have here stage 1, that's the longest stage. So we know the longest stage, that cells spend most of their time not cell dividing. So I know this is no division. And then I know that this stage 3 here, that's going to be the very last stage. So that's what's going to happen last before the cell completely divides. So I'm going to guess that this is the actual process of the cell splitting into 2. Let's go back to here. Now I don't have a ruler on me, but in the exam definitely try and use a ruler. So stage 1, I said there was no replication going on. And so that means that's going to be this one here. Because here it's saying the cell grows and the chromosomes replicate, but there's no hint there of cell division. And then stage 3, I'm going to tackle 3 next, that's probably the easiest one to look at, is that's the final stage of cell division before forming 2 cells. So that sounds to me like this one here in the middle, where the cytoplasm and the cell membrane divide to form 2 new cells. So you're going from one massive cell and it's splitting off into 2. So that's going to be the last stage of cell division before the cell just starts growing and chromosomes replicate. And then so that means order of elimination, or just last but not least, 2 is going to be 1 set of chromosomes is pulled to each end of the cell. So, next, what percentage of the total time for the cell cycle is taken by stage 1? So, I'm going to go here. It might be a bit hard to see on the screen, depending on how good the quality is on your end. But here there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 pieces. So like 10 pieces of pie is the circles divided into sort of 10 parts. So I'm just going to write 10 on the top here so I know I'm thinking of 10. And then how many does the first one take up? So we've got 1, 2, 3, 4, 5, 6, 7. So it's going to be 7 of these. And if this is 7 of these, then that means that 7 out of 10 is the total. So I'm going to go here and I'm going to just write 7 divided by 10, and that equals 1.7. And to get into percentage, you need to times by 100. So times by 100 equals 70%. So it wants a percentage here. So I'm just these divide independently. So they each produce 2 daughter cells. So it's like 2 times 2, 4. And then on the third day, because nucleus is the whole cell, not the whole cell, sorry, all of the genetic information, so all the chromosomes. It could be a gene. I'm going to stick a question mark next to that. It could be. Or it could be a gamete. I don't think it's going to be a gamete because gametes have... gametes are like sperm or eggs. Cells used for reproduction. These are not cells used for reproduction because we're looking at pieces of genetic material, not a cell. So this is a cell, this is part of a cell, so it must be a gene from order of elimination if you didn't know from the start that the answer was already a gene. So stem cells now. Stem cells are cells which have not yet been specialized to carry out a particular job. So they just do something. They continue basically to just sit there, divide and continue replicating by mitosis, and then when needed they specialize. So bone marrow cells are one example of stem cells. Okay, so we know bone marrow cells are stem cells. Explain how a transplant of bone marrow can help to treat medical conditions. So we've got 2 marks, so we need to put 2 points down. And the first thing that we can write about is treat medical conditions. So I'm going to guess that it's something to do with stem cells, and what stem cells do is differentiate. And then these differentiated cells must somehow treat your medical condition. So what I'm going to write is bone marrow cells differentiate into, say, many other cells. In particular, it's blood cells that these differentiate into, but you don't need to know that. And what these cells do is cells help treat damaged tissue. So what I'm going to say is so they differentiate into many other cells which help damaged tissue by replacing damaged cells. And that's going to be my answer for this one. So you can see here that I've gone bone marrow as an example of stem cells. So they're stem cells. They're not yet specialized. I know then that they probably must differentiate. Then how can that be used to treat medical conditions? Well, most of the time you use stem cell transplants when cells are weak, or when cells or tissues are going to be damaged. So that's when you'd use them. So next, we're going to have a look at another question here. Now let me just double check that the stream is going all right for now. I think the answer to that question is hopefully yes. Yep, that seems to be all right. Nothing's crashing. Brilliant. So here we've got, there are two types of cell division. So you've got mitosis and meiosis. Describe three differences between the processes of mitosis and meiosis. So the first bit that you want to do is be able to know which one's which. And the way I remember which one's which is, I remember that mitosis, I just go mitosis, oh, it's about my toe. So I'm literally going to write my toe. And the reason I write that is because then I know that it's going to be division of cells within my toe. My cells in my toes don't make egg cells or sperm cells or anything of the sort, and that's what meiosis does. So meiosis makes, better word is to use gametes. Yep. So first step is three differences. So the first thing that I think I'm going to talk about is about the fact of how many cells are produced by each process. So mitosis produces 2 daughter cells. Whereas meiosis, on the other hand, produces 4. Produces 4. So that's one point. Another point is if we're producing 4 cells versus 2 cells, one of these will have 2 cell divisions, one of these will only have 1 cell division. So if that is the case, then that means that our cells produced from meiosis will have 2 divisions, whereas mitosis will only have 1. So that's something else that we can write about. So you can say there is only 1 cell division in mitosis, but 2 in meiosis. Yep, so that's another point we've put down. And something else that would probably be able to talk about is what are these cells used for? So mitosis produces cells which are absolutely identical, whereas meiosis produces genetically different cells, because these are then used to create genetic diversity, which is then used in fertilization and in diversity of offspring. That's one of the advantages of having sexual reproduction over asexual reproduction, because you have processes of what we call genetic recombination within these processes within meiosis. So that's going to be my last point, where I'm going to say mitosis uses genetically identical cells, and then whereas meiosis produces different cells. So I'm going to write makes genetically almost ran out of space there. Genetically different cells. Alright, brilliant. So, what else can we say? What else could we write about possibly? So, something else that we could say is that in meiosis, we have one chromosome of each pair of chromosomes, whereas in mitosis you have both pairs of chromosomes. So another example would basically was the words haploid and diploid might mean something to you. So mitosis produces, I'm going to write this on the top because I don't have space down the side. I'm going to write diploid cells, and meiosis I'm going to write haploid. So humans have 46 chromosomes in total, but they have 23 pairs of chromosomes. So in mitosis, you will have 46 individual chromosomes, whereas in meiosis, you will only have 23 because you only have one member of each of your pairs. Right. So our next step is going to be describe one similarity between the processes of mitosis and meiosis. So one similarity, which is always going to be the same, you could talk about either cells, divide, but that might be a bit simplistic. So what I'm going to go with is that DNA doubles. So there's DNA replication in both cases. Replicated. Oh, that is not replicated, unfortunately. In both cases. Brilliant. So I'm just going to move this a little bit up. Hopefully you can see that all right. So that does the trick. So here, what we have is figure 1 shows the production of sperm cells in humans. So we've got cell A, there's cell growth and copying of genetic material to produce cell B. And then there's cell division, so we form 2 cells, and then cell division again, and then we form sperm cells. Okay, so we're going to have to bear that one in mind as we flip over to the next set of questions. So actually, what I'll do is I'll keep this on the side, so hopefully you can see both rather than me moving it out of the way. So cell A is a normal body cell. So that's here, we've been told cell A, this one here, is a normal body cell. How many chromosomes are there in cell A? So if it's a normal body cell, we know that it's going to be diploid. So if it's diploid, that means it has the normal number of cells, which in turn means that it's going to have a pair of each chromosomes. So whereas in gametes, you have half of each pair. So in this case, what is the actual base number of chromosomes? So can it be 23? Well, 23 from the previous question that we looked at, it could be if it was a haploid cell, but I've told you it's diploid. So this isn't the case. However, you might write this one down if you're thinking about pairs of chromosomes. So there are 23 pairs, but we're not looking for pairs right now. So we're looking for individual chromosomes, and if we're not looking at pairs, I've told you we want individual chromosomes. 23 is the number of pairs, so how many members are there in total? So if we just double 23, we get to 46, so I know immediately that's going to be the answer. Now, 48 is just there to trick you out between noting whether it's 46 or 48. So in case you don't remember exactly how much, it's just there to catch you out. 92 is if you remember 46, but if we double 46, we get 92. So this is going to be times 2. However, what you need to remember is that 46 is not the base number of pairs. 23 over here, that's the base number of pairs. 46 is the number of individuals, so it can't be either of these. Oh. So, what is the mass of DNA in cell E? So cell E, just make sure you can see this, almost. The cell E is this cell over here. So we've got A, B, C, D, and then it goes to E. So this is after 2 cell divisions. So there's cell division 1 there and 1 there. So what we're curious with, or what we've got in question, is what is the mass of DNA in cell E? And the way I'm going to think about that is basically how many divisions has there been. So in cell B, it's just cell growth and copying of genetic material. So cell B has double that of A. That's what I'm going to write there, 2 times A. Cell C and D have just A. So you can write just 1xa. That's because here, copying of genetic material means you've got DNA replication, but there's been no cell division yet. After cell division, each sort of copy goes into different cells. So this first bit here is almost exactly the same, or you can think of it as mitosis, if that's of any help. If it confuses you, ignore it. So here we have 1 copy of DNA from A. And here, there's 1 more cell division, so we're gonna, with each division, division halves DNA. So half of 1 is just gonna be 0.5 times of A. Yeah. So then if we go back to here, what is the mass of DNA in E? Quarter of the mass of A. Well, that's not what we've got written down. So it's not going to be this. Half the mass of DNA in A. Yeah, that's what we've just discovered. So 0.5 is just half. Same mass as A. Well, we know that can't be true because it's a haploid. So you could also write in the bottom here, sorry, haploid, and this is diploid. So that would also be a quick way, if you wanted to and you knew the differences between the two, to figure that out as well. So haploid, diploid, twice the mass. Well, no, that's B. B's twice the mass. So now, what type of cell division produces sperm cells? Binary fission. That's for bacteria. This can't be right. Differentiation. You could maybe argue it, but it's not in this case because it doesn't involve cell division. So there's no division, and we're talking about cell division. Differentiation is not cell division. So that means we're left with meiosis. So that, I think, leaves us to, that leaves us to the end of our questions. I hope that this has been useful. Again, if you have any questions, feel free to drop them down in the chat at any time, and I'll just do my best to answer them when they come through. And yeah, that will be it from me. So I think, please leave us a like if you like, drop a comment if you're sort of enjoying this content, you found it useful and you like more of it. Please let us know if there's sort of any topics that you want us to go over as well, because if there's something that's a lot of people would like to see, we can definitely try and accommodate for that. And please subscribe if you want to see more of this kind of content. We've got a few streams coming over this week. I think every day at 5 o'clock this week. We've got a stream for half an hour on some GCSE content. I'm not sure about the rest of the week, but tomorrow I think we've got a chemistry live stream for GCSE, and they'll be looking at moles, so please do keep an eye if you're interested and you think this kind of stuff would be helpful. But yeah, this will be it from me. I hope you've enjoyed. I hope you found it useful, and yeah, I will. Actually, we just got one question in chat. What paper was that? This was not from a particular paper. I can find out what paper it's from, but it was from a set of questions from Access Tuition. So I'll put that was from some past paper / topic questions on Access Tuition under cell division. So yeah, you can find them there. They usually don't leave links to what papers they're from, but if you want to know exactly, you can. If you search up one of the questions in total, you should be able to find it that way. Otherwise, it is past paper digging, unfortunately. But yeah, hopefully you found this useful, and that will be it from me. Bye for now, and I'll hopefully see you sometime soon. Thank you for coming.